guys. Today we will continue our connective tissue and blood module. Connective tissue is one of the four basic tissues in the body. Connective tissue mainly formed from cells, fibers, and the matrix. Types of connective tissue generally, it may be connective tissue proper, like loose areolar, dense collagenous, reticular, and adipose and elastic. Embryonic as mesenchymal or mucoid, or specialized, which is a special type of connective tissue have special character as blood, cartilage, and bone. In this module today, we will talk about blood. Blood is a special type of connective tissue. Blood contain, we will discuss first the component of the blood in general. Blood like that. This is a sample of blood contain yellow color and blood and red color, which is centrifuged. The yellow color is called plasma. Plasma represent 55% of the blood, and the blood component, which is red in color, blood cells or formed elements, contain 45% of the blood. The plasma formed from water, organic and inorganic compound. Water represent 90% of the plasma. Organic compounds as proteins formed 9% of the plasma. Inorganic compounds as nutrient and gases, which means oxygen and carbon dioxide, represent 1% of the plasma. So generally blood is formed from plasma, represent 55%, and the blood cells formed 45%. Plasma contain 19% water, 9% organic compounds as proteins, 1% inorganic compounds as nutrient and gases like oxygen and carbon dioxide. Formed elements of all blood cells. We have white blood cells, white BCs, which although named leukocyte. Red blood cells are BCs, which although named erythrocytes. And platelet, which also names thrombocyte. Leuco means white, erythro means red, thrombo means thrombosis, which is the main function of platelet. So plasma 55, blood cells 45, white, red cells, and platelet. Blood film. What is the meaning of blood film? Blood film is the method while I could put the blood in an microscopic slide to be examined under microscope. The staining method of the blood is different. Staining method is called Leishman stain. Leishman stain is formed from three components: acidic stain, basic stain, and azurous dyes. Acidic stain, which is eosine, basic stain, which is methylene blue, plus azurous dye. These three components forms the Leishman stain. Leishman stain contains puts in blood in a slide like that to be examined under microscope. This is the slide under microscope of blood film stained with Leishman stain. Represent white areas, which is the plasma, not stained, and red poles or red colors rounded cells, which is the RBCs, and basophilic cells, which is the white blood cells. This is also a blood film, contains RBCs and white blood cells. White blood cells have many White blood cells have many features or many shapes according to the function of the cell. We will talk about white blood cell in another lecture. This lecture we will talk mainly about red blood cells or RBCs. Red blood cells, erythrocytes, RBCs, all of them is the name of the same cell. The origin of RBCs is stem cell in the red bone marrow. So the bone marrow is the source of all blood cells, like RBCs. The shape of RBCs is biconcave, like that. Biconcave cells, concave from above and concave from down. So this is called biconcave cell or biconcave disc. Any abnormality in this shape is called poikilocytosis. So poikilocytosis is abnormality in shape because the normal shape is biconcave. Any abnormality is called poikilocytosis. Abnormality like it may be spherical in shape, like a ball, which is called the spherocytosis. Spherocytosis means the biconcave is lost and the RBCs became spherical in shape like a ball. Another abnormality like sickle cell, like that. This is the RBCs take the shape of sickle cell, which is a disease in sickle cell anemia. So this is the normal biconcave. The two of them is called the poikilocynosis. If there is abnormality, poikilocynosis may be spherocytosis, like spherical in shape, or sickle cell, like that. Okay, the shape also may be changed according to the solution or the blood, if it hypertonic or hypotonic solution. Normally, blood presented in the body is isotonic solution. Isotonic solution, when the RBC is presented in isotonic solution, it will preserve its normal biconcave structure like that. But in case of hypertonic solution, the fluid inside the RBCs will come out and the RBCs shrinks to be crenated RBCs like that. But in case of hypotonic solution, the fluids from outside became enter the RBCs and make it swelling to be spherocyte and ruptured at the end, like that. Again, this is the isotonic solution. RBCs preserve its normal biconcave shape like that. And this is hypotonic solution. Fluid passes from outside to inside, make it swelling and rupture at the end like that. Hypertonic solution, fluid passes from inside the RBCs to outside to be shrinked and be created RBCs like that. So, is RBCs contain nucleus or organelles and why? RBCs has no nucleus or airy organelles. Why? To accommodate more hemoglobin so that they can transport more amount of oxygen, because the main function of RBCs is to preserve the hemoglobin inside it, which responsible for transport of oxygen to body tissues. Okay? Size of RBCs. RBCs diameter is about 6 to 9 micrometer with average 7.5 micrometer. If the size of RBC's diameter less than 6 micrometer, it's called the microcyte, which means small cell. If the diameter more than 9 micrometer, it's called macrocyte, which means large cell. In case there is a wide variation in size, which means the blood contain microcyte and the macrocyte at the same time. This is called the wide variation in the size. This is called anisocytosis. Again, large cell below 6 micrometer is called the microcyte. Large cell more than 9 micrometer is called the macrocyte. And in case of a wide variation in size, it is called anisocytosis. Okay, what about number? Number of RBCs in males, it is between 5 to 5.5 millions per cubic millimeter, and in female it is below males, it is 4.5 to 5 million per cubic millimeter. Why? Because the regularly monthly blood loss in female in case of periods. Any decrease in number below the normal, this is called oligocythemia, which means anemia. And any increase in number above the normal, it's called polycythemia. So the normal, it generally in males and females between 4.5 to 5.5 per cubic millimeter. Any decrease is called oligocythemia or anemia, and any increase is called polycythemia. Oligocythemia or anemia means decrease in number. There is obvious decrease in number in RBCs. In contrast with the polycythemia, there is increase in number of RBCs. This is RBCs which is increased in number above normal, and this is oligocythemia or anemia, decrease in amount of RBCs below the normal in the plasma. So, why the RBCs appears red in color in blood film stained with Leishman stain? What is the cause of red color? Red color because of hemoglobin protein content. Hemoglobin protein content make it red in color, like that. This is the RBCs stained with Leishman stain appears red in color. Content and the color. Content and the color, it contain mainly hemoglobin. Normally, hemoglobin about 14 milligram. It will be acidophilic with pale center and dark periphery, like that. This is RBCs, acidophilic cell with pale center and dark periphery. This is called the normochromic cell, which is the normal RBCs with normal content of hemoglobin. But in case of decrease of hemoglobin below 14 milligram, which is called anemia, this shape will change it to that, with large pale central area, which is called hypochromic. It will be acidophilic, but the pale central area enlarged in size, so this is called the hypochromic cell with low content of hemoglobin. In case of increased hemoglobin above 14 milligram, this is called polycythemia. The cell will appear acidophilic with a stained central area, the pale central area will be stained, and the peripheral area will be deeply stained more than normal. This is called the hyperchromic cell. So again, hemoglobin content in the cell change in hemoglobin content will change the color of RBCs stained by Leishman stain in blood film. In normal cell with normal hemoglobin content, it will be acidophilic with pale central area and dark periphery. In case of low hemoglobin content, which means anemia, it will be acidophilic with large pale central area and dark periphery, which is called the hypochromic cell. In case of polycythemia or increase in hemoglobin content above the normal, the cell will be acidophilic and the pale central area will be stained with more dark or more deeply peripheral area. This is called the hyperchromic cell. Electron microscope of the RBCs, it shows it lacks nucleus and organelles. As we said, it contains no nucleus and no organelles to accommodate more space for hemoglobin that responsible for oxygen transport to another tissues. The cell membrane of the RBCs is highly elastic. Why highly elastic? Making it flexible to pass easily through narrow capillaries, because RBCs passing through the blood presented in blood vessels varies in the diameter from large artery to medium sized to capillaries. Capillaries varies in size, maybe large capillary or narrow capillary. RBCs will pass through all of these pathways, so it have to be flexible in case it pass through a narrow capillary. So the cell membrane is highly elastic to make it flexible in case of passing through these narrow capillaries. Above the cell membrane there is a cell coat. This cell coat contains an important thing, which is called the surface specific antigen that determines the blood group. Our blood group may be A, may be B, may be O, may be AB. This is the blood group. How it is determined? Determined according to the surface specific antigen on the cell coat above the cell membrane of RBCs. Okay, like that. This is the RBCs. It contains a surface specific antigen, A antigen, B antigen, and RH antigen. RH determine if this blood group positive or negative. As we said, A positive, A negative, B positive, B negative. So this RH antigen determine the A or B is a positive or negative. Okay. The RBCs, it has a well-developed cytoskeleton as a spectrin and actin, like that. This is the spectrin protein. This cytoskeleton, either spectrin or actin, responsible for maintain the biconcave shape. As we said, RBCs is biconcave in shape. Any change or any abnormality in this protein, the spectrin or actin, this cytoskeleton, will it change or will play in the normal biconcave shape of the RBCs. So any change in RBCs, it will be named bikilocytosis. Okay. The cytoplasm of the RBCs contain mainly hemoglobin and water, enzymes and few lipids, but mainly hemoglobin. This water responsible for isotonicity of the RBCs. It may be hyper or hypotonic in case of the isotonic or hypotonic or hypertonic media of around the RBCs. Okay, this is the electron microscope of the RBCs. So what is the lifespan and the fate of RBCs? RBCs lived only about 120 days, about 4 months, and then it dies. How it dies, what is the fate of RBCs? The aged or died RBCs are removed by macrophage cell by phagocytosis in the spleen, liver, and the bone marrow. So, spleen, liver, and bone marrow is the organs that responsible for phagocytosis of aged RBCs by macrophage cell after 120 days or 4 months. So, clinical notes that we couldn't transfuse our blood to another one less than in a period less than 4 months. Why? Because the new RBCs does not yet formed. Okay, what is the structure, function, adaptation of the RBCs? What is the meaning of structure, function, adaptation of RBCs? It means that how the RBCs or how the structure of RBCs prepared to do its function. We know that RBCs contain many functions. How the gut creates the RBCs or structure of RBCs to be able to make its function normally. First, the absence of nucleus and organelles permit enough space for hemoglobin because or due to the main function of RBCs is to contain hemoglobin that carry oxygen to the tissues. So there is no nucleus, there is no organelles to permit enough space for hemoglobin to do this their normal function. Second one, the biconcave shape of RBCs provide large surface for gas exchange. Okay. Third one, the well-developed cytoskeleton maintains a biconcave shape as we said by two proteins, spectrin and actin. Any abnormality in spectrin or actin will affect the normal biconcave shape of the RBCs, which lead to a state called bikilocynosis as spherocytosis or sickle cell disease. Okay, the rounded edge of the RBCs facilitate the passage of RBCs in the narrow capillary. As we said, capillary RBCs may pass in a very narrow capillary. So their round edge facilitate the passage of the RBCs in this narrow capillary so easily. Plus it contains elastic fibers. So RBCs contain elastic fibers plus this round edge to facilitate their passage in the narrow capillary easily without interruption. Okay, the last one, the highly elastic cell membrane allow RBCs to pass in narrow capillary as we said. Okay, so again, how the RBCs structure adapted it to make its function normally, which is called structure function adaptation. First, there is no nucleus, there is no organelles, so there is a space or enough space for hemoglobin. Second, the biconcave shape provides a large surface for gas exchange between blood. Third, well-developed cytoskeleton to maintain the biconcave shape of the RBCs. Cytoskeleton means spectrin and actin. Any abnormality of them leads to poikilocytosis. The round edge facilitates the passage of the RBCs, and highly elastic cell membrane allow it to pass in the narrow capillary. So the elasticity of RBCs allow it to pass from the first, and the rounded edge facilitate this passage in the narrow capillaries. Okay? Anemia. Anemia is the clinical application of the RBC cell. The RBCs, by the way, sometimes it's called red blood corpuscle, not red blood cell. Why? Because true cell, to name it a cell, the cell should contain a nucleus and organelles and the cell membrane to name it a cell. But RBCs contain only a cell membrane. It have no nucleus or no organelles. So it is not a true cell. So it is sometimes called red blood corpuscle, not red blood cell. Okay, let's back. Anemia. Anemia means decrease in number of RBCs below the normal. Anemia have many types according to the cause. It may be microcytic anemia. What's meaning of microcytic? Microcytic means the RBC's size is less than normal, less than 6 micrometer. The microcytic usually hypochromic. What is the meaning of hypochromic? Hypochromic means the hemoglobin content of the RBCs is less than normal, less than 14 milligram. So, in case of microcytic RBCs and hypochromic RBCs, the type of anemia is called the microcytic hypochromic anemia that happened in iron deficiency anemia, as the iron, it is an important substance in the pathway of the formation of hemoglobin. So in case of iron deficiency anemia, the type of anemia presented under the microscope with Leishman stained blood film, it will appear microcytic hypochromic anemia. As we said in this slide, the number of RBCs presented low in normal and the cell presented hypochromic. Why hypochromic? Because the acidophility is below than normal and the pale central area is large in size with dark periphery. This is called microcytic hypochromic anemia. The another type is called the macrocytic hyperchromic anemia or megaloblastic. Megalo means large cell. Macrocytic means the size of RBCs larger than normal larger than 9 micrometers. This is called the macrocytic. Macrocytic usually hyperchromic. Hyperchromic means content of hemoglobin is larger than normal, larger than 14 milligram. So this anemia is called macrocytic hyperchromic anemia that occurs due to deficiency of vitamin B12 or folic acid. These elements also is important for formation of hemoglobin. It's presented like that. This slide represents RBCs in Leishman stained blood film. The RBCs is large in number, large in size in contrast with these RBCs, and the central pale area is stained with deeply acidophilic or deeply stained periphery. This is called the macrocytic hyperchromic anemia. Okay, so any deficiency in the important elements in formation of hemoglobin leads to anemia. In case of iron, it leads to microcytic hypochromic. In case of vitamin B12 or folic acid, it leads to macrocytic hyperchromic anemia. And that's it today. Thank you guys for listening and have a nice day.